We're going to continue talking about how we do get new species. We've already discussed allopatric speciation, which refers to species independently evolving once that population gets separated. We also have what's called sympatric speciation, although this is a lot less common. Sympatric means same country. So in this case, we're going to have new species evolving from each other when they're still located in close proximity to each other. There's a number of different ways this can happen. We're going to talk about polyploids. We'll talk about habitat differentiation or habitat differences. And then we can also have sexual selection, and that's going to contribute to this as well. So let's take a look at these different ways that we can get species formed when the individuals are located close together. We talk about polyploidy first. Polyploidy, the word poly means many. So here we're talking about many copies of a gene, or sorry, many copies of a chromosome set. So as we see in this picture here, we can have one set of chromosomes, that would be 1N. We could have diploid, that would be 2N, triploid, tetraploid, there's hexaploids, octoploids, decaploids. The numbers can keep going up and up. So when we're talking about polyploidy, there are multiple, usually more than 2 sets of chromosomes in the species. This is going to typically occur initially because of an error. So it is because of an error that would be in cell division. So perhaps a cell copied all of the chromosomes thinking that it was going to divide, and for some reason that division didn't take place. So we end up with twice the number of chromosomes in the cell that we had initially. This can certainly contribute to new species forming, because once we have differences in chromosome numbers, that is an immediate isolation for an individual. This is very common in plants, and in fact it's thought that 80% of the plants we know of today evolved as a result of some type of polyploid event. We have essentially 2 different types of polyploids that can form. Auto means self, and so these are polyploids that form within a species. So we have the same species, we get a couple polyploids there, and they're able to reproduce with each other. If we look at this picture, this is representing now allopolyploidy. Allopolyploidy, this is where you have different species that are going to produce polyploids that can combine and now produce their own fertile, viable offspring. Keep in mind that if we're going back to that biological species concept, if we're going to get new species, or what we're going to call a new species, it has to be able to produce the viable offspring. So here we're looking at this scenario. We've got 2 different organisms. One has 6 chromosomes, one has 4 chromosomes. Each of them produces a gamete. Well, perhaps the gamete from the 4 chromosome individual doesn't form correctly. So we end up with this odd hybrid that has some weird chromosomes here, because you've got now chromosomes that don't have a homologous pair, which is really important when we have meiosis taking place. So keep in mind that that is going to be necessary for getting viable eggs and sperm a lot of times. So that can be one of the big things that disrupts this polyploid process. However, if we do somehow get this gamete down here and we get this other gamete, when those 2 combine, we could end up with a fertile hybrid over here, which now has more chromosomes than what the original parents had. And every chromosome also has a homologous pair. So this one you would expect is going to be able to go through the meiosis process just fine. We're not going to have any difficulty there. And so that would be a new species, because it's no longer going to be able to reproduce with the original parents, because again, the gametes don't really match, and it would be able to reproduce with other individuals that now have that same number of chromosomes, which in this case is going to be 10. Another example of how we can get an allopolyploid new species, we get these two gametes, they form, we get again an odd hybrid that is haploid. We don't have those homologous pairs here, but if there's some kind of an error in the cell division process, all of those chromosomes could end up with a homologous member. Once that happens, then the rest is very simple. We get meiosis, we get normal gametes, which are able to combine with gametes just like themselves. So again, we have kind of this instant forming of a new species because you have that instant reproductive isolation. Habitat differentiation is another type of sympatric speciation. We can get new species formed even though all of the individuals in the population are going to remain in close proximity to each other. Kind of a common way that this happens is with food preference. Sometimes some individuals in the population will just begin to prefer a new food source. If we look at this example, we've got a fly population. Suddenly some of them prefer green apples and some of them prefer red apples. Okay, they're still very close to each other, but we're not going to get as much mating between the two populations because they're just kind of hanging out with other flies that like the same food as them. So what you get is decreased gene flow between the two separately preferring populations, and eventually you get enough changes or differences between the two that they no longer are going to mate with each other at all. And then eventually they're not even able to do so. Here with cichlids, we have another example. They have different food sources. This might mean that they then start to primarily inhabit slightly different places around, say, the coral reefs, so that even though they could reproduce with each other, they just don't because they're more in contact with similar preferring fish. This has been very important for cichlid speciation in some of the lakes in Africa, where we have just thousands of cichlid species that have evolved all right there in close proximity with each other. Another thing that's contributed to cichlid diversity, and then also some other species, is sexual selection. So when we talk about sexual selection, we're talking about basically mate choice. In this picture here, you have two different looks in the population, and they begin to prefer one look over another. It's not to say any one look is better than the other, but there's this preference here. So we start to get blue females choosing the blue males and the green females choosing the green males. And so they are somewhat drawing a line down the middle, even though they are still located right there next to each other. They're living with each other on an everyday basis. They're just not reproducing that way. So this operates right against Hardy-Weinberg, as we discussed in chapter 23. Once you start violating those principles of Hardy-Weinberg, you do get divergence of the population. So we start to see some evolution and some changes in the allele frequencies. Eventually, it becomes enough so that we do get complete reproductive isolation of those two groups, and they no longer can reproduce with each other, so now we call them a new species. Sometimes it's just a slight coloring on the feathers that a female starts to prefer over another color. It may be a slight different color on the fins, a different shape. It may not take very much to cause the females in the population to start to prefer one versus another. Okay, so what if we end up with some reproductive isolation, but not all the way reproductively isolated? This is very common. So keep in mind that speciation takes time. So you may get some species that do prefer one mate over the other, or one mate look over another, but maybe there's not a whole lot of those that they are preferring. So we get a lot of hybrids formed. So when we have incomplete reproductive isolation, we get what we call a hybrid zone. So a hybrid zone is a zone where we have hybrid or in between or intermediate individuals. So as we see in this picture here, we have a single population that has a certain amount of gene flow that's taking place. So we expect that we're getting random mating, no natural selection taking place, but something happens so that we do get a barrier to gene flow. This could be a change in mate preference. It could be a change in food preference. Something happens though, so that we have this difference. Once we have this difference, we've interrupted gene flow so that two groups in the population begin to evolve independently from each other. If those two populations are still breeding with each other a little bit, you get what's called the hybrid zone. This hybrid zone is individuals that are intermediate to the two somewhat distinct populations. And there's three different things that can happen with the individuals in this hybrid zone. The first outcome is that we could get reinforcement of the two distinct species or two distinct groups so that they are able to form into distinct species. This might happen because those hybrid individuals, if we're talking about something like sexual selection, perhaps they're not so desirable looking. So even though we get formation of the hybrids, maybe those hybrids are not going to have the opportunity to reproduce because they don't have the desirable look to them. In that case, we just get more separation of the gene pools in the two different distinct species. We could get fusion of these two distinct species. So although they somewhat separated from each other, we may end up with so much gene flow that they do start to meld back into one kind of homogeneous population. The other thing that we could get is we could just get stability. Maybe the hybrids are able to mate, but they don't mate that much, so that you still have hybrids in the middle. You still have your two distinct populations kind of on the outer rims of all of this. So we do have hybrid zones. Those hybrids may be fine. They may stay there. We may get just so much separate evolution of the distinct populations that the hybrids eventually stop forming altogether. If we talk about how fast these new species are going to form, there's a couple different things that have been observed from the fossil records. It's been observed where you have the sudden appearance of new species, and that's what this first top picture is representing, where you just suddenly see this new look to an organism in the fossils. That's what we call the punctuated formation of species. That's certainly been observed before many times. And when we say it's a fast appearance of a new species, we're talking about something like maybe within 50,000 years. Okay, so fast is not 5 years, it's not 10 years, it's not really 200 years. But fast, we might see a new species come just in a matter of 50,000 years in the fossil record. We've also observed what we see on the bottom here, which is the graduated formation of new species. So there may be a more gradual change in the species. We can just see the slow molding of the species into this new morphological phenotype. Again, we can have both situations take place. It has been observed. One is not necessarily better than another. And either way, we have to keep in mind that evolution is working on the phenotypes and those phenotypes' interaction with the environment. And it may be an interaction with both non-living things in the environment and then with other living organisms that are present at the same time.